Animal research. Even the phrase evokes controversy. Through your training, you know that animal research has led to key medical advances and has made an enormous difference in the length and quality of our lives. As a physician, you hold a unique and influential place in the community. Friends, neighbors, patients, and others look to you for guidance on complicated and sometimes controversial scientific issues. The American Medical Association is calling on its members to promote, enhance, and defend biomedical research. Americans for Medical Progress offers this presentation to give you the tools and resources to do just that.
There is a growing threat imposed by animal rights organizations and activists to restrict or even eliminate biomedical research. This will impact not only you and your practice, but the patients that you care for. It's important for you as a physician to understand the mistruths and misinformation provided by these organizations on what biomedical research is, what animal research does, and the impact on the future of medicine. Animal rights organizations have tried to influence animal research in a number of ways, through legislative means, through state and federal regulations, through education, through demonstration, through media attempts as portrayed on movies and in television, and in some cases through criminal acts. This is all in spite of the fact that the majority of Americans, and certainly the majority of physicians, support the use of animals in research and understand the need of animal research in the advancement of medicine.
Hi, I'm John Young, a laboratory animal veterinarian and director of the Department of Comparative Medicine at the Cedars-Sinai Medical Center in Los Angeles, California. As a laboratory animal veterinarian, I work with physicians and other scientists who use animals in medical research to enhance our ability to prevent, diagnose, and treat disease in human and non-human patients. In my 2.5 decades as a laboratory animal veterinarian, I've come to know that most physicians understand the vital role that animals play in medical research. However, it's also become known to me that most physicians do not talk to their patients or the public about the vital role that animals play in medical progress. As a result, many members of the public, particularly patients, fail to make the connection between the use of animals in medical research and increased ability to prevent, diagnose, and treat disease. There is a small minority of individuals in this country, leaders of animal extremist organizations, who would, if they could, eliminate all use of animals in medical research. And these leaders have taken advantage of the failure to the public to understand how and why animals are used in medical research to misrepresent and vilify the use of animals in research in the eyes of the American public. The philosophy of the animal extremist group leaders is actually quite simple. All animals are sentient, and all animals share the same rights. Human beings share no special rights, and any use of animals on the part of humans is speciesism, the moral equivalent of racism and slavery. Ingrid Newkirk, president and co-founder of the animal extremist organization PETA, People for the Ethical Treatment of Animals, succinctly summarizes this philosophy, and I quote, "A rat is a pig, is a dog, is a boy." Tactically, the animal extremist philosophy is publicly played down and is not used in their solicitation materials or their campaigns of misinformation. Instead, the leadership of the animal extremist community tells the 6 anti-research lies. 1, the use of animals in research is inherently cruel and always painful. 2, the use of animals in research doesn't work because animals aren't people. 3, the use of animals in research is unnecessary because, on the one hand, almost all disease is preventable through lifestyle changes, or even if some research has to be done, there are viable alternatives to all uses of animals in medical research. 4, the use of research animals diverts huge amounts of money away from direct patient care. 5, pets are stolen and used in research. And 6, the use of animals in research is so morally reprehensible and those who engage in it so evil that violence and terrorism perpetrated against those who use animals in research is justified. While the animal extremist group leaders speak in sound bites, unfortunately for physicians, medical research is a complicated subject and is not easily explained in sound bites. However, there are three simple truths about the use of animals in medical research that patients, the public, and the media needs to hear. One, in the past century, virtually every medical advance depended heavily upon the use of animals in medical research. Two, for the foreseeable future, the use of animals in medical research will be vital to medical progress. And three, without the use of animals in medical research, we cannot maintain the status quo. Without using animals in research, medical progress will slow, halt, and eventually reverse.
Many of the treatments and procedures that you provide your patients today that many of us take for granted were in fact developed in animals using animal research. Some examples: diabetes. Insulin was discovered in dogs. Surgical procedures such as heart valve replacement, the heart-lung machine, organ transplantation, orthopedic prostheses, neurosurgery, chemotherapeutic agents that we use to treat patients with cancer, and many examples where animal research has benefited humans. Without animal research, these treatments would not be available to our patients. My particular area of interest is in transplantation immunology, trying to understand why transplanted organs might reject or be accepted. We use animals that are genetically altered, transgenic animals, in order to study specific pathways that might lead to rejection or tolerance. Without these animal models, it would be very difficult, if not impossible, to make advances in the area of organ transplantation.
Since most physicians have not been inside of a facility where animals are actually used in medical research, and since a picture is worth 1,000 words, let's take a look inside the facilities at Cedars-Sinai Medical Center. The coronary arterial anatomy of a pig is almost identical to that of a human being, so they're very good models for testing out prototype coronary devices, like arterial stents. Also, the size of a farm pig of about this age, which is somewhere around 4 to 6 months, very closely approximates the size of a human heart. So they're actually anatomically and physiologically very good models for cardiac device testing. Hey, big pig. We're going to go in this room next where we have dogs with a prototype device for the heart to measure intra-atrial pressure, which will greatly assist physicians who are treating heart failure patients. These dogs have a device implanted in them called the heart pod. The heart pod will, through telemetry, measure in real time intra-atrial pressure non-invasively, allowing the physician to better monitor his or her patients. These mice are actually being used in the Neurosurgical Institute laboratory. The Neurosurgical Institute is looking for better ways to treat a very aggressive brain cancer called glioblastoma multiforme. So the Neurosurgical Institute is looking at ways to improve the ways to diagnose and treat this disease. These mice are actually nude mice. They're mutant mice, which is actually a naturally occurring mutation. They are athymic. They have no thymus or T white blood cells. Therefore, they are immunocompromised. When we have a mouse like this, we can actually administer human glioblastoma multiforme and study the human disease actually in a mouse model. So our researchers here have 9 different lines of human prostate cancer and 2 lines of human breast cancer. And when pharmaceutical companies discover promising new anti-neoplastic drugs that work in vitro, they then contract with researchers at Cedars-Sinai Medical Center to actually test the drugs in vivo. So we have the prostate cancer cells, the breast cancer cells inoculated underneath the skin of these mice, and these mice then receive the new potential anti-neoplastic drug to check for safety and efficacy. So these are so-called knockout mice. They've had a gene knocked out that codes for high-density lipoprotein, so they are very sensitive to atherosclerosis when fed a high cholesterol chow. They also have been intranasally inoculated with chlamydia pneumoniae. And these mice are being used to unlock the relationship between chlamydia pneumoniae infection, development of atherosclerotic plaque, thrombolus, and emboli, which actually cause fatal disease in human patients. These mice, of course, are specifically bred for research. We feed them pasteurized food. The bedding that they are housed on is autoclaved. The water they drink is highly purified. It's 18 megaohm ultra pure water. And essentially in the cage sanitation facility, after these cages are washed at 180 degrees plus detergent, they're bacterial culture negative. These mice have all been inoculated with a virus called herpes simplex virus. It's a human pathogen. You have it, I have it, virtually every human being does have it. We're inoculated soon after birth, and the virus in most people lives dormant in the brain, in the trigeminal ganglia. In most people, when the virus reactivates, it travels down the facial nerve and causes a cold sore. However, in a large population of people, for reasons unknown, it migrates down the optic nerve and causes an infection of the cornea. This is one of the most leading causes of blindness of children in third world countries. The ophthalmology department at Cedars-Sinai Medical Center is trying to develop a vaccine that will prevent the reactivation of the herpes simplex virus in the trigeminal ganglia as a cure for permanent blindness. Our researchers at Cedars-Sinai are partnering with veterinarians at a major veterinary school in the United States that has a large oncology center which sees dogs with spontaneous glioblastoma multiforme. So as I speak, we are conducting clinical trials in the veterinary school on dogs with spontaneous glioblastoma multiforme. If we can cure those dogs, just as we've cured these rats, then the Food and Drug Administration will give approval for us to use this gene therapy on our human patients. So we're selecting certain genes we know that are in triplicate in Down syndrome babies, genetically engineering these mice so they actually have these genes, and then doing very intricate tests on behavior and development to actually learn which specific genes in Down syndrome contribute to mental retardation, abnormal brain development, and cardiovascular pathology. This rabbit is being used to study sudden cardiac death, which is a disease we see in post-myocardial patients where they have atrial and ventricular fibrillation, which actually in most cases is fatal. In the human condition, information is rather limited. We can look at autopsy specimens and try to postulate as to what caused the fatal fibrillation, post-myocardial infarction, and the sudden cardiac death. But this rabbit is a very powerful model to help us actually learn what is happening in real time. This rabbit was taken to surgery and under anesthesia had a myocardial infarction induced, which the rabbit tolerates quite well. Of course, the animal is under anesthesia, and we're very aggressive with post-operative analgesic therapy. This facility is our quarantine facility for zebrafish. The zebrafish that you see in this facility are either wild types or mutants, and the mutants are genetically modified to actually be predisposed to have abnormalities of spinal cord and brain development. And the target for the researchers here is to unlock the mysteries of the genetic basis of pituitary tumors. This is our simulation laboratory. In this laboratory, we can actually teach new surgeons basic surgical techniques without using animals. Now, if you look around the room, you'll see endoscopic, laparoscopic simulators, intubation, bronchoscopy simulators, colonoscopy simulators. These, however, are not true alternatives to the use of animals. They are adjuncts. They allow the young physician, the young surgeon, to learn basic surgical and practice technologies in principle. But these phantoms have no blood, they don't have real tissue feel, and they really don't simulate the living animal or the living human patient. So these simulators actually allow us to decrease the number of animals we use in teaching and training, but they don't allow us to completely eliminate the use of animals in teaching and training. As a laboratory animal veterinarian, I've been engaged in public outreach to explain to people how and why animals are used in medical research for almost 2.5 decades. In all that time, I've only been asked simple variations of 10 questions about how and why animals are used in medical research. I'm now going to share those questions with you and give you my brief responses in the hopes that you will take it upon yourself to help the American Medical Association demystify the use of animals in medical research and explain to the American public how and why animals are used.
Animals are so different from people. How can we learn anything from animal research?
Just because there are differences between the human and non-human animal does not mean that there are not similarities. And it's part of the job of the laboratory animal veterinarian to assist the physician and scientist in selecting the appropriate species for study. For example, mice share 95% of their DNA with us. Therefore, mice make very good models of genetically caused diseases. Dogs and pigs have cardiovascular systems and gastrointestinal systems, which are very similar to humans. But it's not always the similarities between humans and animals that are the focus of study. Often it's the differences. For example, sharks rarely, if ever, get cancer. Amphibians can regulate their blood pressure at will, and cockroaches and salamanders can regenerate entire body parts. If we can study these animals and learn how they perform these medical miracles, perhaps we can apply what they do to the human condition.
Are these animals lost pets, or are they stolen?
There's approximately 90,000 dogs and cats used annually in the United States in medical research. Greater than 86% of these are bred specifically for research and by definition cannot be lost or stolen pets. The other small percentage of animals are sold by so-called random dealers, which are regulated by the United States Department of Agriculture. The USDA tells us that they successfully trace back animals that are sold by random source dealers greater than 97% of the time.
Why do physicians and veterinarians participate in research that harms animals?
Physicians, veterinarians, and technicians participate because they realize the use of animals in medical research is a vital cornerstone to medical progress. The veterinarians and technicians have the animal welfare as their priority. We are the advocates for the animals. We participate to ensure that medical progress is ensured while the animals are treated as humanely as possible.
Do the animals suffer? Are they in pain?
There are veterinarians and veterinary technicians involved in research on the front line. We are the advocates for the animals. We ensure that research is done in the most humane way possible while medical research is facilitated. The use of anesthesia is routine, as is the use of post-operative analgesia. The use of animals in research is highly regulated. Federal law requires the search for alternatives to potentially painful procedures.
Why do animal tests continue when cruelty-free products are available?
The cruelty-free marketing campaign is a legacy of the 80s, when in the cosmetic industry, Avon, Revlon, and Mary Kay Cosmetics charged a premium for their products while calling them cruelty-free. In reality, the corporate definition of cruelty-free was different from what the American public perceived. According to the corporate lawyers, what cruelty-free actually meant was, we didn't test the product for safety in animals in our facility, we paid someone else to do it, or we took compound A, which we had tested for safety in animals, and compound B, which we tested for safety in animals, and put them in the same bottle, calling it compound C and not testing it for safety. By law, whether it's a cosmetic or a pharmaceutical drug, it must be tested for safety and efficacy before exposing it to human beings.
What happens to the animals after the experiments?
At the end of the experiment, in almost all cases, the animals are euthanized, or put to sleep. And the American Veterinary Medical Association dictates the most humane methods to use. Now, why are the animals put to sleep? It's not because we don't want them anymore, we don't care about them, or don't want to provide for their care. The animals are euthanized because in almost every instance, in order to answer the questions being asked, we must harvest the organ system or tissue of interest and examine it microscopically and at the molecular level. If we don't do the microscopic and molecular examinations, the animals are wasted. Not all animals do require euthanasia, and for those that don't, most facilities have adoption programs.
Why does the number of animals used in research keep going up?
For most species of animals, over the past 50 years, the numbers of animals being used has decreased from 20 to 30% due to refinements in the experimental methodology. However, in the case of mice and fish, the numbers of animals are on the rise because they are very powerful tools in genetically based research, the so-called transgenic and knockout technology.
Why can't you use alternatives, like computers instead?
Computers only model known phenomenon. We don't know how the brain sees. We don't know how digestion works. Therefore, we cannot make computer simulations to simulate those physiological processes. The most sophisticated computer software program in existence today pales in comparison to the complexity of a single liver cell.
Do we have the right to use animals? Do the animals have rights?
Animals have the right to be treated humanely, but physicians and scientists have a moral obligation to use the knowledge and the tools available to them to enhance our ability to prevent, diagnose, and treat disease and minimize suffering in human and non-human animals. The use of animals in medical research is a vital cornerstone to medical progress, and to involve human beings in potentially harmful research is illegal. Previous data in animals demonstrating safety and efficacy is required by the Food and Drug Administration to allow human patients to be enrolled in clinical trials. Now that you've heard the most frequently asked questions about the use of animals in medical research, you're prepared to speak to your patients about new treatment and diagnostic modalities that came to fruition through the use of animals in medical research. When speaking to your patients, use simple, clear language, not technical terms. Use numbers, percentages, and statistics, but not too many. And practice your message points. When speaking, show empathy, confidence, and sincerity. Be positive, comfortable, and honest. And above all, don't be defensive nor become intimidated. Do not become emotional or confrontational.
I hope the preceding information has helped you understand the need and the role of animal research in clinical medicine. I hope also, as a physician speaking to another physician, that I can persuade you to become an advocate as a spokesperson for your patients and others that you might work with in an encounter on a daily basis, to help them understand the importance of animal research in the future of medicine.